welcome to podcast.init, the podcast about Python and the people who make it great. I would like to thank everyone who supports us on Patreon. Your contributions help to make the show sustainable. When you're ready to launch your next project, you'll need somewhere to deploy it, so you should check out Linode at www.podcastinit.com/Linode and get a $20 credit to try out their fast and reliable Linux virtual servers for running your next app. Do you need to learn more about how to scale your apps or learn new techniques for building them? Pluralsight has the training and mentoring you need to level up your skills. Go to www.podcastinit.com/pluralsight to start your free trial today. You can visit the site to subscribe to the show, sign up for the newsletter, read the show notes, get in touch, and support the show. To help other people find the show, please leave a review on iTunes or Google Play Music, tell your friends and coworkers, and share it on social media. If you work with data for your job or want to learn more about how open source is powering the latest innovations in data science, then make your way to the Open Data Science Conference, happening in London in October and San Francisco in November. Follow the links in the show notes to register. Your host, as usual, is Tobias Macey, and today I'm interviewing Peter Koch, Wibowo Arindrarto, and Tiago Antão about Biopython, a suite of Python tools for computational molecular biology. Peter, why don't you start by introducing yourself first?
Hello everyone, my name is Peter Koch. I started using Python back in about 2003, 2004 when I was introduced to it by a lecturer when I was doing a master's. And then during my PhD, subsequently, I began using Biopython and started contributing and eventually ended up as one of the main developers on the project.
And Bo, how about you?
Hi everyone, my name is Bo. I've been officially using Python since 2010, I think. I started actually working in the lab, as in a proper biological lab, but then I started gradually shifting towards a more programming role, and now I'm a scientific programmer in working in the Netherlands. But my contribution to Biopython, I think, officially started around 2011, if I'm not mistaken. That was my first commit, I think. But it's still going on up to today.
And Tiago, how about you?
Hello all, my name is Tiago Antão. I'm currently a research scientist at the University of Montana in the US. I started using Python around 96. My background is slightly different. I came from computer science to biology, whereas most folks, I think, go in the other direction. I did my first commit to Biopython. Peter might even remember better, but around 9 years ago, I think.
So to start off the conversation, I'm wondering if one or all of you could start by explaining what bioinformatics is and highlight some of the different areas of research that are held under that umbrella.
So a lot of us joke that bioinformatics is 90% file format conversion. We have lots and lots of computational tools that are designed to solve biological problems, but there tends to be an invent-your-own file format mentality, and there are rival file formats doing similar things but slightly differently. So a lot of day-to-day work in bioinformatics is file format manipulation, and things like Biopython can help with file format parsing. So that's one of the strengths and one of the reasons many of our users use Biopython and related tools.
And so it sounds like Biopython is primarily a glue between the multiple different data formats that are used by researchers and people who are doing the analysis in the field.
It's a fair summary. There's a lot more in Biopython beyond that. We have things for dealing with three-dimensional protein structures. We have clustering code for more numerical work. But what a lot of people use, and certainly what I use, you could describe as glue, yes.
And do any of you know the full history of how Biopython first got started?
Not directly, so it was started before any of us three were directly involved. Do you remember the year, anyone?
I think 1998. Yeah, 1998. That was way back.
Back in the early days of bioinformatics, a lot of the work was done using Perl because systems administrators were being roped in to help with projects, and they at the time preferred and used Perl. But there are other languages, and people preferring Python started wanting to do bioinformatics in Python, and that's how Biopython really started. The same for sister projects like BioJava and BioRuby that came along a little later. So BioPerl is, I think, very slightly older than BioPython. But we started, about late 1999.
And so given the long history that Biopython has, I'm wondering how the project has evolved over that time to meet the changing needs in terms of both the research that's being conducted and the computational landscape that's available.
On a personal level, we've had lots of people come and go. It tends to be that as people get promoted, they no longer have the time to dedicate to the projects, whereas new trainee scientists coming in are more likely to be able to invest a considerable amount of time. So there's a turnover in personnel. And as you say, technology has moved on. The Python language itself has moved on. We're now on Python 3. There's been a lot of maintenance work, cleanup work to transfer us over from Python 2. And interesting new technologies as well, like making sure that Biopython works on PyPy. I'm sure some of your listeners will know about PyPy. Runs your Python code and optimizes it as it goes. PyPy is an interesting project.
Another thing that I would like to raise is how the ecology of software on the Python side changed a lot. So if you were doing this in the late 90s, you would essentially, your software package would be mostly working alone. Whereas nowadays you have a massive ecology of software, of very good software that you can interact with, say NumPy, SciPy, scikit-learn. So it's more in terms of the ecology of software. It's more like it's teamwork with many other packages. Whereas at the beginning, it was really just a small isolated project with very few scientific libraries or charting libraries. So in that sense, I think it has been a massive revolution in terms of the whole Python ecology experience.
Yeah, I guess since if it's for 90, I think you can say that the Python ecosystem has changed a lot. I think that there's so much more there's so much more Python libraries that you can use, and more specialized ones to process various data these days. But if you look into the Biopython project, you can actually see that there are still, I wouldn't say remnants, but you can clearly see that this is a library that has been developed quite over a long period of time. I think the development started even before PEP8 was introduced, sort of like the de facto standard. That was a long time ago. I think Git was not even around. I think the original versioning was done using CVS.
Yes, CVS.
So that was the era in which the Biopython project was started, I guess. But now it's completely in Git. It hosted in GitHub, and then we're modernizing part of the libraries a lot. So we're it's been going on for a while.
And on the science side, there have been plenty of changes too. From my point of view, the biggest has been what we call high throughput sequencing. Yeah. More and more DNA and RNA data coming through. And some of this is so high volume that you need specialist optimized programs to deal with. But again, there's still plenty of glue work for interoperability.
Yeah, indeed. I think maybe this also hints to one part of the library that was, well, it's not the major part, but we do have some C code, right? We do have some highly optimized functions that are written in C that the users of the library can interface with Python. But that's also becoming more common these days. People write the parts that need a lot of performance in C, and then you write a Python wrapper on it, and then you access via Python functions, just as in a regular Python script. So.
A big point, I think it's exactly that. So it has become a big data discipline. I know this is too much of a fancy word nowadays, but in this case, is very true. So if you were working 10 years ago, a floppy disk would probably have all the data that you could that you would need. But nowadays, we are talking about tens of terabytes of data that need to be processed, cleaned up. And it becomes something that it needs the most out of your computer skills to analyze the data that we have from DNA sequencers.
And I'd say that that's probably a pretty large impact on the overall bioinformatics field, is the recent capabilities of being able to do gene sequencing and more complex analysis of the actual underlying biological material.
Yes, that's been a major change, but there's plenty going on top of that, and things like the visualization plugins in various international databases and online APIs for connecting resources. So plenty of change in many areas.
I would just like to make a point, probably should be made some time ago at the beginning, but bioinformatics is a field that different people can do completely different things. So what I specialize in my case, is population genetics, which is completely different from what Peter does, and it is completely different from what Bo does. It's not just that we are dealing with different biological species. We are dealing with very different kinds of analysis tools and different and different problems, and that makes some of the work that we do, the specialized computational biology work, very different from user to user, and there are different areas in bioinformatics. So some people analyze, compare species across species. Other people look at specific genes. In my case, I look at data across populations of the same species, and so on and so forth. And there are people that work with proteins, and it's a massive field. And the ones of us that do the most, some of the research, actually have very different skill sets on the biological side.
And I imagine that must manifest as a bit of complexity in the Biopython project, trying to be able to cover such a broad range of requirements.
Effectively, it's an umbrella of submodules which would have had an original author and maintainer. So Tiago is in charge of the population genetics part of Biopython, for example, and it's semi-independent from the rest of Biopython, and that kind of works for the most part. I mean, the stuff that I do on the sequence side tends to be more connected because sequence data crops up everywhere.
How does Biopython compare to the sibling projects implemented in Perl and Ruby and Java?
So the ones you've named, BioPython, BioPerl, BioJava, and BioRuby, are all of a similar age and have overlapping community membership. So those four together set up what we call the Open Bioinformatics Foundation, which initially was a non-profit company to look after the servers and domain names. It's now still a non-profit, but it's part of software in the public interest. And we still have domain names, but now with less physical servers. It's mostly Amazon stuff. But anyway, there are other projects using the same naming scheme, like there's a BioGo, and I think someone's trying BioJulia. There's a BioJS project. They have different biological areas of strength. Some are very broad, like BioPerl and BioPython. Others are much more focused. So BioJulia, I think, is very strong on the visualization side. But they're not exactly competitors. We're all trying to do research in biology, and we will contribute ideas and test cases, and it's a fairly positive attitude, not like the tongue-in-cheek rivalry between fans of particular programming languages. At least that's the attitude I try and take with this.
I think in most major programming languages today, you can find like a de facto bioinformatics library indeed. But I think this also reflects the amount of, and the type of people, I guess, working in bioinformatics, because you can easily find Python programmers, Ruby programmers to an extent, people who are familiar with Java and everything. And if you're coming from different backgrounds, I think it's not that difficult to find a library that you can work with, or even you can start contributing to.
So for somebody who's using BioPython, what does a common workflow look like when they're working with biological data?
Touches on an earlier part of the conversation. Most people are doing very, very different things. Also, workflow has many meanings in the field. It can mean an automated workflow, like a shell script or a YAML file or an XML file that describes a reproducible, rerunnable, repeatable pipeline for an analysis, the kind of thing that you might include in a scientific write-up of your work. But I think there's no one-size-fits-all answer for what a typical day of bioinformatics analysis looks like.
Unless chaos is an acceptable answer, but there is lots and lots of variation, as Peter is saying. So it's completely different from user to user, and that sometimes is problematic, because one of the things that we are very concerned is about a replication of results. And sometimes it's a bit frustrating that things are too ad hoc-ish in a way. And I think it's worthwhile referring here a project which is the Galaxy project, which is implemented in Python 2, and which is an amazing project that allows precisely to have reproducible and standardized workflows.
So what are some of the most interesting or innovative uses of BioPython that each of you are aware of?
That was probably the hardest question on your list. It crops up a lot behind the scenes. Good infrastructure doesn't get noticed until it breaks. And I think a lot of people use BioPython or BioPerl or whatever as infrastructure just to get something done. It isn't in itself flashy or exciting. So I struggle to come up with a good answer. I'm hoping one of my colleagues on the call can come up with a nice example.
I also cannot pinpoint an exact answer. I think, well, maybe several, but what I remember is that when I started writing some libraries, I started getting some emails from people and then they say, well, I've been using this script in-house. We've been using this your module in our web service, and can you actually make this fix for us? Can you make this feature for us? So it's not really prominent per se, as in you don't see that immediately, but once you touch on the libraries, when you work on some feature, you get usually some in contact with people who are actually working on this. Again, I think this reflects on the various subfields or the various different parts of bioinformatics that people are working on.
I can actually give you some examples of applications that I've worked with, and some very, very practical applications. For example, we have used Biopython and Python to help in the Yellowstone National Park to make decisions about bison and about culling of bison, for example. Obviously, there is a big part that is slightly, it's in the Python world, but not BioPython per se. It's about simulating genetics and trying to have measures of viability of populations as a function of their size. And then you can make management decisions on populations, and very, very practical decisions were made with regards to the sizes of the population of Yellowstone bison. Another example is, again, in the field of population genetics, to trace the path of diseases or resistance to diseases. For instance, you can use BioPython and Python and these kinds of population genetics tools to trace that resistance for malaria, which is still one of the biggest killers in the world, tends to start in resistance to drugs. Tends to start in Southeast Asia, and then mostly to expand from there to the rest of the world. Another example is when you get all this data from sequencers, and again from the field of malaria, you can you can use all these methods and all these tools. Again, it's not just BioPython; it's the whole ecology of bioinformatics software to, for example, see what parts of the genome are influenced by, in the case of, say, mosquitoes that transfer malaria, which parts of the genome are influenced by are creating resistance to insecticides. So with that information, then you can, for instance, see how effective a certain insecticide is and what effects is having on the mosquito genome. So these are very, very practical applications that the infrastructure is there, and you need this infrastructure to help answer some of these problems.
That's a nice high-level answer from Tiago with some high-profile examples. I was thinking more little things like people on the mailing list or on Twitter or on Stack Overflow where they've had a problem and they've had a short solution from a community member using BioPython. That kind of thing happens a lot, and I like that. So little steps too.
Yeah, that's definitely one of the great things about open source software and open science in general is the ability for cross-collaboration that can have compounding effects on the quality and capabilities of the end result. So what are some of the most challenging aspects of developing and supporting BioPython, both from a technological and a community level?
The very mixed environment that people run this software in. We try and support Windows and Linux and Mac. We occasionally have people on FreeBSD and other things as well. That has definitely got a lot easier in the last 10 years because of publicly minded companies like Travis CI or AppFair that provide essentially free continuous integration testing for open source projects. So we now have automated testing for the Linux platforms and for Windows. And that's really great because typically our developers will, for example, work on Linux and they won't have easy access to a Windows machine. So if a user says something's breaking for me on Windows, it's not always straightforward to work out why. So that's one challenge. Another we've touched on is the age of the project. Some of the bits of the code are very old and the person that originally wrote them are no longer actively involved. Over time, we've updated things. We've done some spring cleaning, and we're trying to improve the unit test coverage, which helps in that situation greatly.
If I may add something on the technological level, I think it has also again to do with the fact that we parse various file formats a lot. And a lot of the specification of the file formats are actually not so well defined. And some of them are actually meant to be human readable, but they contain so much information that it's still useful to be able to parse. So we write parsers, but then we still see some corner cases for specifications which are either not well defined or not so well followed by people that the parser breaks, and then we have to come up with add a new test case and then try to parse that so that it works. But then it's we have a very big list of unit test cases, I think, for this to exactly to tackle this problem. So it's been a challenge on that part, I think.
As an example of that, in the age of Unicode and different encodings, one of the more recent problems is strange characters in data sets where the encoding is not explicit. So should we interpret this as an eacute, or is it invalid data, or what? And occasionally this happens even with big public databases where they are curating user-submitted information and strange characters get in. And it could have been someone wrote this up on a computer set to, I don't know, a Russian keyboard or whatever, and this character has entered the database, and when you try and pass it on an American setup, it doesn't work. So Unicode will solve this, but at the moment, bioinformatics has plenty of legacy file formats, and encodings is one of the gray areas that's completely underdocumented.
And another problem is the fact that some of the people developing some of these tools, they just learn to program in whatever language to develop that tool, and it's very common that you see some. For instance, the most strange thing I've ever seen was a supposed XML file which had comments, not XML comments, but comments in the middle, and it was anything but a standardized format. And it's very common that people that do tools, they are essentially researchers, that they are really not programmers, and dealing with some of the formats and some of the problems of those formats can really be challenging, and sometimes not in a good way.
And what are some of the most exciting developments in bioinformatics and biology that have either happened in recent memory or that are sort of in development that each of you are keeping an eye on?
Personally, I would say the revolution in sequencing is really exciting, and it's still changing. Currently, sequencing machines give you quite small fragments of DNA, and they've got longer. They're now into hundreds of base pairs, but the current latest sequencing technology should give us even longer reads of DNA, and this will change a lot of the analytical workflows. Before, it was like a jigsaw puzzle where you had to put together the tiny pieces to get the answer. The new sequencing machines are going to give you much larger jigsaw pieces. They're going to make certain tasks much easier. And also these machines, the Oxford Nanopore in particular, are very small. They're portable. And I think we're going to reach the point where perhaps in 5 years, you will be able to sequence things literally in the field with your laptop hooked up. And that will open up an awful lot of interesting biological applications. For example, population monitoring, disease outbreak monitoring, agriculture, medicine. I think that's one of the most exciting developments in biology and bioinformatics at the moment.
Yeah, not much to add there. It's really the big thing, and where sequencing technology goes, we will have to follow. And some very interesting applications are probably just around the corner.
Yeah, I have to add that I have the same thing written down on my notes, that the nanopore is probably one of the most exciting thing that's happening in recent times. You get much bigger sequencing length, or much bigger jigsaw puzzle pieces, as Peter mentioned. But I'd like to add that there is that's on the technology side. So that's the part where the data is generated. On the practical side, on bioinformatics, actually, I'm actually have been and it's still excited by the development of the Python ecosystem. I think I have a feeling it's becoming the de facto language for large parts of bioinformatics, and it's one of the things that I'm interested in is actually how people are coming to a more standard way of structuring their data pipelines or structuring their workflows. So this is also has been a problem, I would say, in the field. So aside from having so many different file formats, we also have different ways of saying how a pipeline should run. But the community is coming together now more closely now to agree on a standard and to say, this is my set of data analysis pipelines, and you can run it on so many different backends, but this is how I define it, and everybody can understand.
Probably another exciting development is, because lots and lots of data is being generated, is the fact that high performance computing is becoming a thing, and development of high performance algorithms is now very important. And suddenly biology, that was like not using much, suddenly it needs a lot of expertise, or being on the forefront of fields of everything related to cluster computing, GPU computing, etc. And this is probably going to increase and increase and increase as people are able to to generate more data in a cheaper and cheaper way.
It's interesting from a point of view of the next generation that biology undergraduate students now are starting, or have been for some time, having to learn not just mathematics for statistics, but also programming. Many universities now include substantial programming courses for biology students. So it's becoming a more numerate field, and that is exciting and important for the future.
Yeah, I also have the impression that in the field you can find everybody from, I mean, it's the big umbrella is biology, but you can find people easily from computer science, biologists themselves, physicists, chemists, all sorts of statisticians. Everybody's coming together, and it's really reflected in the development, I think, now and in the coming years. So a lot of exciting things.
And one thing that I'd like to go back to is, given the increase in the availability of sequencing data, and I'm assuming that that's going to increase the overall size of the data sets that you need to process. Yes. So I'm wondering if you foresee any difficulties in the length of the sequence, creating any additional complexities in processing it within Biopython, and whether that might require any sort of architectural or just sort of refactoring in those sequencing libraries.
One of the main tasks with DNA and RNA sequencing is genome sequencing. And the interesting thing there is that as the technology improves, this should actually get easier. So it wouldn't surprise me that this is effectively a solved problem, and we wouldn't have to worry too much about dealing with lots and lots of jigsaw pieces to put together a genome. But yes, input-output is a bottleneck, and Python isn't always the best fit, but then you can drop down to C or call into specialized command line tools for doing the analysis. Things like sequence lengths can be a problem. Some of the early file formats and tools in this area used a 32-bit limit on index sizes, and that works okay for humans, but doesn't work very well on plants or even other animals like marsupials. So the nitty-gritty of the computer science does get involved, and sometimes is handcuffing us.
So for somebody who wanted to get involved in Biopython and start contributing, how much domain knowledge is necessary for them to get started? And are there any areas of the project that would benefit from somebody who's purely focused on the software engineering aspects of it?
Let me just say something. I ended up in population genetics essentially so I'm a computer scientist by training. So when I started this, I did a master's. I ended doing the course on population genetics by mistake. So it can really be overwhelming. And I ended up doing population genetics now, but it can really be overwhelming, and the terminology and everything can be overwhelming. My suggestion is don't be put off by that. Don't be afraid. And because currently there are many things to do, and some of them are a bit tedious, like all this glue is not probably the most exciting stuff, but if you like that's great. But there are lots of exciting stuff and occurring on the algorithmic side, because this deluge of data needs to be processed in an efficient and reliable way. And if you might need to understand a little bit of the problem, but after that it's mostly programming and statistics. And I would say that more than the biology, sometimes statistics can be, especially when you are developing algorithms, statistics can be even a bigger bottleneck in terms of knowledge than even biology itself. But sure, there is lots of space nowadays for people that are interested in the field, and they can actually just do very interesting programming and computing and work on cutting edge stuff just to process this deluge of data, which is data that is ugly, that has errors, that needs to be corrected in very sophisticated ways in terms of statistics and programming. Because the data that we get from sequences has errors and there's lots of copies, and that makes for very exciting problems. It's not just glue, but plenty of other stuff that if you are able to get through the biological jargon, you will find really interesting stuff on the programming side.
So for people wanting to contribute to the project, I would normally ask them what area of bioinformatics are they working on, and try and match them up to a particular bit of BioPython that matches those strengths. But we have people with very little or no biological background, and there are still plenty of things that they can do. If they have, for example, knowledge of C, they may be able to help improve our C bindings, or even what you might call housekeeping work, like style improvements to better match the PEP8 style guidelines, or doc strings, improving the documentation in the code. There's plenty of things that people can do without necessarily having any biological or technical domain knowledge. So a variety of contributors is great.
And one of the things that I'm curious about from your answer, Tiago, was when you do have errors in the sequencing data, I'm wondering if that's ever due to actual mutations in the genome and what some of the difficulties are in differentiating between legitimate mutations versus bad sequencing.
That is a serious problem. But generally, no. The problem is that the sequencer, because we are dealing with very, very small structures, chemical structures, it's the sequencer that's not sure about the reads, and what the sequencer sends some metadata, which is normally the quality of the read that the sequencer thinks it has. And if you are actually, one of the interesting applications is trying to find honest mutations, real mutations. And generally speaking, the amount of it depends on the organism, but in most organisms, the mutation rate is much, much lower than the error rate. So the two things conflate themselves, and you need to spend lots of time trying to find what is a real mutation versus what is an error. Again, especially because error rates are normally much, much higher than real mutation rates. But I think both Peter and Bo probably have something to contribute here, because they probably work or even more than me on that subject.
I think it really depends on the quality of the incoming data, as in the raw biological sample, also the machine itself, because there are actually a plethora of machines today, and they all have different error rates, but also different trade-off, of course. But I have the impression we can never really say up to absolute certainty. This is where the statistics come in, actually. So you have to model these things into your calculations. You have to model the error rate. You have to model the maybe the underlying mutation rate, and then you come up with an answer, and you actually you test your hypothesis that way. But it's something inherent so far, I think, that needs to be taken into account in all the downstream calculations.
And occasionally the error is actually from the computer. This isn't a Biopython example, it's an example from an assembly tool called Mira, and the author had a weird bug report. So DNA sequences are represented as strings of letters, and for some reason he was getting the wrong letter, and he couldn't work out where this mystery letter was coming from. It didn't match the input data. He eventually tracked it down to faulty RAM, so a cosmic ray or just chance had flipped a bit and had changed the letter, and it was a problem in the computer itself that had corrupted the data. So there are plenty of things that can go wrong, and checking your code and your pipeline for positive controls and negative controls to make sure that what you're seeing is real is an important part of science on a computer.
And what are some of the most problematic limitations of Biopython and the overall bioinformatics landscape, and how do you work around them in your day-to-day?
I would say inconsistent file formats is one of the most problematic things, and test cases and wider user uptake is one of the best ways to work around that. If more people are using code, the better exercised it will be, the more likely the corner cases will be found and reported and fixed. But against that, we have a rapidly changing ecosystem. The sequencing technology in particular is changing rapidly, and the file formats and standards themselves are evolving over time. So it's a moving goalposts, and to me, that's one of the biggest limitations. So user feedback is how we can really work around that.
I have to agree on the file format. I think that's one of the I don't want to use annoying as the word, but it's very early in the analysis that you encounter these things sometimes, but you really have to take them into account. And you really, if you want to make a script or you want to make a program that can work in most cases, you have to take them into account. And I think that's there's no way of knowing what kind of error that's going to occur other than doing analysis and expecting people to actually report back to you if they found something weird or unexpected, and then coming up with a way to circumvent that.
As an example, gene names, the people that discover a gene usually get to name it, and normally the names are fairly sensible. They're made up of letters and numbers, but you get special characters in there as well, like minus signs or angle brackets or quotes, and those tend to cause problems when they're embedded in larger file formats.
It's actually usually a nice, well, I don't want to say nice again, but I think it's sort of good in a way that if your program errors out when it cannot parse something, because there are also some problems. For example, there are many ways to represent a coordinate in various file formats. Some people use 0-based, some people use 1-based accounting, but these things don't trigger an error. And sometimes when people don't conform to a file format that it should be 1-based, but they use 0-based, for example, and the analysis just goes through until the end, and then you don't notice the error until very late in the analysis process. So that's something that also needs to be in the back of most people working in bioinformatics today.
And that's the optimistic view, because I suspect many errors are actually not detected at all because they are silent bugs, as Bo was saying. So I wouldn't be shocked if many of these analyses with important silent bugs actually get published and nobody really detects the problem there.
Yeah, that's a bit worrying indeed. And I think to this extent, we actually do a lot of validation in Biopython, right? I think a lot of the code is checking whether, sanity checks, whether the file formats that we're parsing is really what we think it is.
A good portion of the bug reports are people with weird files that don't follow the format, but something's gone wrong. So yes, Biopython tries to be quite strict on parsing things and catch errors early. Well, that's a design choice.
Mm-hmm.
Yeah, in general, it's definitely better to fail fast and fail loudly so that people can account for that and maybe potentially provide some override path if they say this isn't actually an error, it's something that I'm legitimately trying to do. Mm-hmm. But yeah, being able to raise issues earlier in the process will definitely save a lot of time and effort.
Resist the temptation to guess, which is a philosophical difference. I think, for example, BioPerl, for their file format parsers, they are more willing to do things like automatically detect the file format, whereas in Biopython, we said, look, the user should know what it is, and if it doesn't match, that's usually a sign that there's a problem somewhere else.
Yeah, I think that's also alluding to one of the Zen Pythons, I guess. Better be explicit than implicit, I think.
Exactly, yes.
So for people who are interested in getting involved in the field of bioinformatics, what are some of the bits of encouragement that you might offer, and maybe some reference material that they might look at to determine if that's a path that they want to go down?
The advice I give people is that they're going to have to learn to program, and the best choice of your first programming language would be whatever your peer group is using, so that you have people right there in the room that you can talk to and get help from. So if the group happens to work on Python, great, start learning Python. If they all use Perl, then learn Perl. So in-person feedback when you're getting started is really helpful. Once you build up some experience, you're going to be able to help yourself with online searching and Googling for error message. That is a very practical way of learning to program. And unfortunately, you need to do that in bioinformatics as well.
I think the answer will vary slightly with regards to your background. So, for instance, if you come from computer science, you might be overwhelmed by all the biology and all the biological problems. And again, I think it's important that you find probably mentors or somebody that can help you navigate what is the enormous world of biology, and you really need to have a conversation with researchers, which by the way probably need you, because there is a massive need of people to do data analysis, and finding good mentors on the biological side is very important if you come from computer science, I think.
Yeah, I cannot overstate the importance of having mentors, be it like direct mentors that you have in your school or in your maybe your lab, also something that's probably online that you don't meet in real life very often, because there's just so much happening in bioinformatics and in so many different subfields, like on the mathematics part, on the statistics part, on the technological side, on the computer science, the algorithms that people write to analyze this data. And you can easily get lost with so much development. You don't know which one to pinpoint to, and you don't know which one to pay attention to more closely. So I think, having a mentor, having a peer group is probably the most important thing. And I would also say to add to resist the temptation of writing everything your own, and maybe that's a good way to learn, I think, if you want to do it on your own. But there's a lot of the tool set, a lot of libraries are getting mature, and then it's not that difficult for these days to find a collection of people or a collection of libraries that have been developed, taking over so many corner cases that you're probably not aware of, that you may have stumbled if you had written your own tool. So that's again the importance of peer groups, I guess, because they can pinpoint you to the latest advances and the latest best practices to grow in the field.
And just to note also, there is some asymmetry in information on the internet. So if you come from computer science, you are used that you Google for everything, and there is an answer. There are actually 10 answers, and documentation is great. But if you then go into the research side, the amount of available documentation is much, much lower, and the communities are smaller. So it's much more difficult if you don't have somebody to help you and guide you, because the amount of available information that really helps you is not as good as if you go in the other direction. Because as we all know, if you want to learn programming, actually, if you are at home, you can actually, there's lots and lots of stuff. But if you want to learn cutting edge biology, there is much less stuff around.
It's one of the nice things about being involved in Biopython and the wider community here. It's networking and meeting other like-minded people who may be able to help in practical things about how to do a particular analysis, what tools to recommend, or job opportunities, that kind of thing. I like the community here, and that's one of the reasons why I'm still actively involved many years later.
So are there any other topics that you think we should cover before we start to close out the show?
I'll probably just add that if you come into this field, you have to like multidisciplinarity and people with different thought patterns, because I imagine most listeners here are programmers or relating to programming. They'll have to learn a little bit of biology. They'll have to learn a little bit of statistics, and they might end up interacting with biologists, statisticians, biochemists, and which have different mentalities and different ways of addressing problems. And that's really, if you like that, that's really awesome. But you have to be prepared that this is a multidisciplinary effort where you will sooner or later have to learn new stuff that you are not very comfortable with.
So for anybody who wants to follow the work that you each are doing and the work that's being done on Biopython, I'll ask you each to add your preferred contact information to the show notes. So for anybody who wants to follow up, they can do that from there. And with that, I'll move us to the picks. And my pick this week is a conference that's local to where I'm at in the Boston area. It's actually happening up in Lowell, Massachusetts. It's called Keep It Low Conf, and it's a DevOps-oriented conference. So it's fairly small scale and low key, but it's trying to reimagine some aspects of the sort of common tech conference and make it a little bit more, I guess cozy is probably the best way to describe it. So for anybody who's in the area and interested in that sort of general field of technology, I definitely recommend going and taking a look at that. It's run by one of the local community members, so definitely worth supporting his efforts there. And with that, I'll pass it to you. Do you have any picks for us today, Peter?
I'm going to pick Jupyter Notebooks, formerly known as IPython. For those that aren't aware, roughly speaking, it's a way of combining software and notes, so markup languages, in one document, a bit like Mathematica or MATLAB. So you can have your figures and your code for an analysis all in one place. And it's very nice as a teaching tool or for sharing an analysis with a colleague. And there are various things online that will exploit this. So if you just stick them up on GitHub, then it will render them in a read-only way, which is really neat. And I want to play with these more.
And Bo, do you have any picks for us this week?
I think I'll go with Conda, actually. That's sort of like a distribution channel for scientific packages these days. It's very similar to PyPy in that I think it takes a lot of inspiration from PyPy. But it's actually becoming more used in bioinformatics today because it can deploy not only Python packages but also other binary packages that are used often in bioinformatics. There's a bioinformatics specific Conda channel called BioConda. If you want to distribute your packages, especially ones related to bioinformatics, I encourage everybody actually to look at it and how they do things. It's not perfect 100% reproducibility, but it is very close and it's very convenient to use, and it's very convenient to also deploy packages there. So I think that would be my today.
Okay. And how about you, Tiago?
I have a few. So if you want to develop in the JavaScript world, the Brighton project allows you to write Python 3 directly on the browser. And it's really an interesting way of getting your feet wet in the web programming space. If you are a Python programmer. On a non-technical note, if you want to know where Paradise is, that is Glacier National Park and Flathead Lake in northwestern Montana, and it's probably the most beautiful place I've ever been, and I encourage you to visit it because it's if you like nature, it's absolutely mesmerizing.
Well, I appreciate all of you taking the time out of your day to join me and talk about the work you're doing with Biopython and bioinformatics. It's definitely an interesting field that's seeing a lot of activity and innovation, so it's definitely a space I'm interested in keeping an eye on. So I appreciate your time, and I hope you enjoy the rest of your days.
Thank you, Tobias.
Thank you. Bye-bye.
Thank you for organizing this.